Okay guys, Life Sciences Grade 12 Revision Classes Term 1 Week 2. My name is Mr. Mpepu and today's date 17 September 2024 and the time there 11:30. With that being said guys, Anbonani, Dumelani, Afsheni, hello, Nda guys. If you are new here, don't forget to subscribe, don't forget to hit that notification bell on. And then if you are a returning student guys, thank you for supporting this movement. Thank you for choosing me, thank you for being here. Yesterday we managed to deal with our week 1 revision. Today we'll be at week 2 and some of week 3 because we're also going to cover the RNA in the location and stuff, right? And then the DNA profiling will be here today. So in less than 10 minutes or 10 minutes, so let's see if we can cover everything, right? So thank you for being here. Those who are new here, while they're subscribing, those who are returning student, while they're liking, while they're commenting, let's move on guys. Are we ready? I'm also ready. Let me timer. Yesterday we managed to reach here, so today we'll start here, right? So let me put on my timer. My timer is on. So thank you guys, thank you for being here, thank you for choosing me. RNA, ribonucleic acid. Yesterday we did DNA, deoxyribonucleic acid. We only have 2 types of nucleic acid in our body, RNA and DNA. But RNA also consists of nucleotides, right? Let me pen. RNA also consists of nucleotides, right? And these nucleotides, they have nitrogenous bases found in RNA are adenine, uracil, guanine, and cytosine, right? Different from DNA here. In DNA, we see thymine. In RNA, we see uracil. The most different that we, if you know this, you know better. RNA contain uracil, DNA contain thymine. From there, we see the 3 types of RNA with messenger RNA, ribosomal RNA, and T transfer RNA, abbreviated as follows: mRNA, tRNA, and tRNA. mRNA carries the code or the message, they carries the code for protein synthesis from DNA to the ribosomes, right? And then RNA forms ribosomes, which are the site for protein synthesis. And then tRNA brings amino acid to the ribosomes to form protein, right? As you can see, we're also going to do this tomorrow during our protein synthesis. Then we'll be talking about this tRNA and mRNA. So let's move on. But there are 3 types of RNA: ribonucleic acid, with messenger RNA, ribosomal RNA, and transfer RNA, abbreviated as follows, right? So let's move on. But located, where are they located, this RNA of ours? The messenger RNA, they are found in nucleus, but then enters the cytoplasm where it attached to the ribosomes, right? Still remember yesterday we talked about the nucleus and the nuclear pore. This is where the mRNA leaves our nucleus to the cytoplasm, right? And then we have ribosomal RNA. This one is found in the ribosomes, and then where in our cytoplasms, right? And then transfer RNA, we found this freely moving in our cytoplasm, collecting amino acid and bringing them to our ribose, right? But the structure of RNA, also like DNA, RNA also consists of monomers, nucleotides, which link form a chain, right? A polymers, right? And then this RNA, this is a single-stranded structure. A single-stranded structure. DNA is a double helix structure, right? Which is not coiled. And then the sugar in this one, it's a ribose, and then it's attached to the nitrogenous bases. You know what is a nucleotides, and then we know what is a nitrogenous basis. The phosphate and the sugar molecule, they attach together alternative to form a chain. As you can see here, this is an illustrative. A phosphate is attached to a sugar, phosphate, sugar, forming a chain going down, right? We have the phosphate sugar, we have the ribose sugar, we have the phosphate group, we have the ribose sugar, because this one is RNA, and we also have the nitrogenous basis, right? And then all this one here is a nucleotide, right? As you can see here, the difference between RNA and DNA, this RNA contains uracils there. But there are 4 types of nitrogenous bases. We have adenine that match with our uracil, and we have cytosine that match with our guanine, right? Here, as you can see, not thymine as in DNA, right? As you can see, move on. The role of RNA, we have did them up there, messenger RNA to take the code or to transmit the code from DNA to ribosomes, ribosomal DNA that forms the ribosomes, and then the transfer RNA that carries the amino acid to a ribosomes, right? All in all, playing a very important role in protein synthesis, right? And then we can compare between DNA and RNA, but we can have the similarities and we can also have the differences, right? The similarities, both of them, they contain sugar alternatively with our phosphate, right? We see a phosphate, we see a sugar, we see our phosphate, we see sugar, right? And then they contain the nitrogenous bases. Both of them, they contain adenine, guanine, and cytosine, and then both of them, they play a role in a protein synthesis. If we don't have DNA, we are not going to have a protein synthesis. If we don't have RNA, we are not going to have the protein synthesis, right? And then we also have the differences as tabulated below. DNA contains deoxyribose sugar. RNA, we see that the sugar is ribose sugar there. And then DNA, it's a double helix structure, and then RNA, it's a single stranded, right? And then in DNA, we see a nitrogenous base thymine, and then in RNA, we see a nitrogenous base uracil, the most different between the two. And then from there, the DNA is found in nucleus only, and then RNA is found in nucleus, ribosomes, and a cytoplasm, right? This one here, we also found them in our nestra nucleus, but this one here, this DNA that we found in our nucleus, also it's the one that plays role in our protein synthesis, right? Then by DNA replication is a process whereby one DNA makes an identical copy of itself. This occurs during the interphase of a cell cycle, right? You know that a cell cycle, before the cell cycle, we have the phase interphase, right? So this one, it can be classified in our steps, right? Step 1, step 2, step 3, step 4, step 5. In step 1, we see that our DNA double helix, it unwinds, right? From unwinding, it's going to unzip, right? From this unzipping, this is where our weak hydrogen bonds between the nitrogenous bases are broken. As you can see here, I create the DNA. You see that the DNA is like this, and then this DNA is going to unwind. After unwinding, it's going to be straight like this, and then from there we see an enzyme breaking these weak hydrogen bonds, forming two strands, right? And then these strands, they are going to work as a template, right? A template is just a something that is going to each original DNA strand serves as a template on which its complementary strand is built. A template is a foundation, and then as you can see from there, we go to our step number 4, where a free nucleotides build a DNA strand onto each original DNA strand, attaching the complementary nitrogenous bases. As you can see here, this was our strand, and this was our strand. As you can see now, our nitrogenous bases, they are being attached to our old strand. And then at the end of the day, we have two identical DNA molecules. This one made out of and each molecule consists of one original strand and one new strand. This one can be an old strand, and this one can be also an old strand, and these two being a new strand, right? As you can see, both of them, they are identical. You can see them, both of them, they are identical. And DNA replication is important cell division, particularly mitosis. It allows each chromosome to copy to each an identical daughter cell, and then as we can move on, so that both daughter cell can have a identical DNA, right? And then we also have the errors that occur during DNA replication. Here we see that if the error occurs in DNA, this can lead to a mutation, right? And as you can see, if the correct nitrogenous base attached to a original strand or a nitrogenous base is added or deleted, we end up with a new DNA molecule, and this results in a change in a gene structure. And then as you can see, we have activity there, DNA replication. There's the structure, read the structure. So you can see this one was double helix, and then from double helix is going to unwind. With an unwinding, is going to be broken down, and then from the broken down, as you can see, the new nitrogenous bases or the new nucleotides with nitrogenous bases are being attached to our complement or to our template, right, forming two identical DNA. Name the process illustrated in the diagram. DNA replication. State the significance of the process mentioned, and then so that during mitosis, during the cell division, both cell can have identical DNA. Identify parts, label 1, 2, 3, 4, 5. I'm wasting my time here. 1, it will be sugar. Number 2, it will be phosphate group. Number 3, this one will be nucleotides. Number 4, this one will be the nitrogenous bases. Describe how this process take place. You can describe up there 5 steps. Give one location of an extranuclear mitochondrial DNA, right? From there, we can move with mitochondria, with chloroplast. From there, we can move to our DNA profiling. DNA profiling, this one, it is made or it is produced by an X-ray film. This pattern consists of lines which are different lengths and thickness and in different position, as shown in the figure below. An individual expect the identical twins have the unique DNA profile. So if we are identical twins, we are going to have a similar DNA profile, right? As you can see here, we have individual 1, individual 2, individual 3, and from this we can conclude that individual 1 and individual 2, they might be relative, right, because of the same location of our DNA profile, right, or our strand or our bar there. Then what DNA profile are used for? To identify crimes, to prove paternity, to identify relatives, to establish a complementary DNA during organ transfer, and to determine the probability or the cause of a genetic defect, right? So this one you also know. And from there, but DNA profiling is a general accepted as a being extremely reliable. The interpretation and the comparison of profile should, however, be approached with caution for the following reasons. Because here we use human to interpret, which makes the mistake. They can make a mistake, and then the method of profiling is different from laboratory to laboratory. Only a small piece of DNA is used, so profile might be not 100% unique to a particular individual, and the DNA profile is expensive, right? The DNA is expensive, therefore not really accessible for those who cannot afford it. And then the DNA profile may be reveal information such as HIV and other abnormalities, right, that can lead to covering person, but the insurance can say, because you are HIV positive, we cannot cover you. So you see. And then we can have an activity there, and then activity we can see the victim, and then we can see the crime scene sample. We can have suspect 1, suspect 2, suspect 3, suspect 4. From the victim we can see that these bands here, yes, they are from the victim, right? So now we see the bands that are correlating to our crime scene. We can see that this one wrong, this one is wrong, this one is wrong, this one is wrong, this one is wrong. We exclude suspect number 1, suspect number 2. We can see this one yes, this one yes, and this one yes, right? So suspect number 2 can be our suspect here in this crime, right? Because the binds they are at the same location. So that was it. My time is up. Tomorrow we'll be dealing with protein synthesis, and then but which suspect probably injured the victim? We said number 2. Give the reason for your answer. Because the bands are at the same location, list one application for DNA profile other than solving the crime. Relative identity, to identify the relatives. Give 2 reasons why DNA profiling may sometimes be challenging. It's expensive, and we also use human to identify or to interpret this DNA, this profile, right? So thank you for tuning in. Thank you for being here, guys. Also, as we don't forget that we are fundraising. You can donate 10 rand, 20 rand, 5 rand, whatever the amount that you can donate, from my numbers 0711906168. Until we meet again, let's meet tomorrow. Protein synthesis. Peace.